This is Screen Capture Video Unit 12.1, the first video of the series. It has to do with the chemical basis of heredity. And when we talk about the chemical basis of heredity, we're talking about the information that's stored in DNA, how it's stored, and then how it's transferred to other chemicals and eventually to be read and used by the organism. This follows Chapter 10 in Biology by Campbell et al. And in talking about this unit, I mentioned in a previous video about previous genetic work. One of the things that we see in science is in order for us to study what work has been done, somebody had to go to the effort of carrying out experiments and recording their work, and for those recordings, those writings to be passed down through history. And so we don't have a lot of that in ancient history. Most of our history is more recent. But I mentioned when I was at the university, one of my instructors, a botany instructor, actually mentioned the first genetic experiment being found in the book of Genesis. And I looked it up. It's chapter 35 if you want to take a look. It has to do with Jacob's cattle and sheep and the arrangements that he made with his uncle Laban for his two daughters. He worked 14 years for them. Some think that while he was out in the fields, he would have watched the goats and the sheep and the cattle breeding and noticed the offspring. And that's something that people naturally do when they're involved with artificial selection or breeding of animals. And even with humans, we tend to look at offspring, sons and daughters, and try to compare to the parents the physical similarities. And you may think that goats aren't that colorful, but I pulled some pictures off the internet, and these are some goats that have different color patterns to them. And it gives the idea that with the different color patterns, this is something that would be due to genetics, because the physical expression of an organism is due to the genetic information that is held in DNA, deoxyribonucleic acid. Here's another goat. I have several pictures of these just to show that there are different types of goats. I've talked about Mendel in the past. Mendel lived in the 1800s and was studying the pea plants. We studied him in Unit 11. I won't have a whole lot more to say about him, but he is given a great amount of credit for the early genetics, the early genetic work. He purposefully did studies with pea plants, and because of his math background, science background, he applied his math and his statistics to the work that he was doing with the pea plants and the different characters of the pea plants: flower color, the size of the flowers, the peas, whether they're wrinkled or smooth, and the color of the peas, and so on. He studied those, and he kept good records, and he passed those on. And so many people give him credit with being the first geneticist. And he was a clergyman in the Roman Catholic Church, so he had support where he could do studies, and he was educated. And as I may have mentioned early in the semester, if you go back before 1900, most of the scientists were somehow connected with clergy because they were learned men. They had a rich education, especially for that time, usually spoke several languages, and they had the support of the church so that they weren't having to work every day to stay alive, toiling in the fields. And this is a statue commemorated to him. There's a guy by the name of Griffith, and in the early 1900s, I believe it was the 1920s, 1928 comes to my mind, but the specific date isn't all that important. He worked with mice and bacteria, and he was actually working with the bacteria, and he was using mice as the host for them, infecting the mice with different types of bacteria, and watching what was going on, and he discovered what was called transformation. I have several illustrations that show what he did. He took living virulent, and the virulent would be the disease-causing, the dangerous bacteria. He would inject that into the mouse, and he'd find that the mouse would die. That made sense. He would take living avirulent bacteria, and these happen to be Diplococcus pneumoniae. They have been renamed Streptococcus pneumoniae, and if I could show you pictures of the bacteria taken of them through the microscope, and you understood what strepto and diplo meant, you'd understand why the name change occurred. But decades ago they made a name change, so if you go back and look up the bacteria, it's now called Streptococcus pneumoniae. It used to be called Diplococcus pneumoniae, and there are at least two different forms of it. One forms a capsule, and so the colonies look glossy, have a glistening appearance to them. And then the other strain does not form capsules, and the colonies appear dull. You took the avirulent, and the prefix A means not virulent, strain of bacteria, injected those into the mice, and the mice lived. Everything seems good to go here at this point. So then he took the heat-killed virulent bacteria and injected those into the mice, and because they were heat-killed, the mouse lived. That sounds reasonable, what one would expect. But what he found peculiar, it was new to him, was that if he took the living avirulent bacteria and the heat-killed virulent bacteria and injected both of those into the mouse died. And when he analyzed the tissue, he looked at the bacteria and looked at the colonies. He found that the bacteria that were recovered were living virulent bacteria. And so the question arises, where did these living virulent bacteria come from? Because the virulent bacteria he injected were dead. What was living were the avirulent bacteria. And so he decided that there was a transforming factor. This is back before DNA was understood to be the genetic material, years before. But there's a transforming factor that could move from the dead bacteria into the living bacteria and cause these living bacteria to become like the dead bacteria. Transforming factor is called transformation. Here's another illustration, and it illustrates the same type of thing. And I even have a third illustration that I pulled off of the internet. You're welcome to take a look at those. In the early 1950s, Hershey and Chase, I think it was Alfred Hershey, Martha Chase, were doing experimentations with their teams, and they determined that DNA was the genetic material and protein was not, because up to this point in time, it was understood that proteins were complex, three-dimensional, complex, large molecules, and it might be that because of their complexity, they might be the genetic material. Well, their experimentation in the early 1950s pointed to DNA being the genetic material and protein was not. They used radioisotopes, and the radioisotopes that they used were one of sulfur and one of phosphorus. And they used radioisotopes because in using radioisotopes, they could take pictures and detect the radioactivity. And they could detect the radioactivity based on location. You'd actually have to see the atoms or molecules, which they wouldn't have been able to do at the time, but they could detect the radioactivity. They worked with viruses called phage, and that's an important biological term. A phage is a specific type of virus that only infects bacteria. And each type of phage is specific as to which bacterium it attacks. This appears to be an illustration of a T4 bacteriophage that would attack E. coli. They're showing the membrane of the bacterial cell. They're not showing the entire bacterial cell. This virus attaches to the surface and it injects its DNA, and these are DNA viruses, injects its DNA, which is the genetic material, into the cell. That's our understanding of it today. Some viruses are RNA, but those that infect bacteria would be DNA viruses. When I first saw this, when I was in my undergrad years, It reminded me of a spaceship that was sent out to the moon back in the late 60s. So those are called phage, and the DNA is the genetic material that's injected. The rest of the phage is made up mostly of protein. That's important to remember. DNA versus protein. It's possible that the protein can be labeled, and here it's shown in yellow because of the sulfur. They used a radioisotope. Protein is made up of sulfur, has sulfur in it, so if they used a radioisotope of sulfur, the protein would glow, so to speak, at least by photographic image. They could use the radioisotope for phosphorus and label the DNA, because DNA contains phosphorus but not sulfur. And again, protein contains sulfur but not phosphorus. This is how they kept them separate. So only the protein was labeled with sulfur, and only the DNA was labeled with phosphorus. With that information, then, they had broth that they grew bacteria in. They released the phage into this broth, gave them time to inject their genetic material, whatever it was. At this time, they weren't sure. Then they put them in a blender and shook off whatever was on the outside of the bacterial cells. And they took the fluid and they put it in a centrifuge tube, spun it, and then looked at it, and they found that the radioactivity, sulfur, which would be in the protein, was outside the bacterial cells. This pointed to protein not being the genetic material, because the genetic material needed to be inside the cell. They then took bacteria and grew them in broth. They put in phage that had incorporated radioisotope of phosphorus, let them attach, inject their genetic material, put them in a blender and shook off what was on the outside, and put it in a centrifuge and looked, and the radioactivity was inside the cell, which is where the genetic material should be. So from this work, using the radioisotopes, it was determined that protein was not the genetic material, but DNA was the genetic material. So this directed scientists in the right direction of the choice between protein or DNA, and they started to follow DNA. And the next question would be, what was its structure? How was it put together? Excuse me. And just so you have a little bit of background on this, the phage attaches to the bacterium, injects its genetic material, and today we understand it's DNA. The DNA of the phage redirects the activities of the cell, and it tells the cell, make more phage parts. These phage parts are assembled, phage are produced, they break out of the cell lyses, that means it breaks down, it's destroyed, and these phage can then reinfect other bacterial cells. This whole process, this cycle, is called lysogeny. When you think of lice, you think of destruction. Okay, just a little bit of a review. We understand that DNA is made up of nitrogenous bases, thymine, cytosine, adenine, guanine, recognized by the letters, the first letter of each, T, C, A, and G. Not so concerned that you know the pyrimidines and purines at this point, but an easy way to remember that, I think of ag, pure, we like pure food, and that's an easy way to remember that adenine and guanine are purines. Sometimes it helps to think of little things to help remember, otherwise it makes it more difficult. We remember that DNA is a double-stranded helix. The sugar phosphate groups form the backbone and are on the outside of the molecule. And here's a model, and you can see the blue, yellow, blue, yellow, blue, yellow repeating, winding around the molecule. They keep the color scheme fairly consistent. And the nitrogenous bases are situated to the inside with hydrogen bonds joining them. And of course, the coiling of the molecule helps its stability. In the next video, I'll continue on a little bit, talking a little bit more about DNA and some more characteristics of it.